Here we go. Hello, everyone. Hello, and welcome to Alta Live. My name is Beth Spotswood, and I am the digital editor of Alta Journal. Today, I am so excited to speak with our guest, Ben Novak, about his work, what we're calling Inside California's Frozen Zoo. The topic today is really just about the conserving endangered animals and how that's being done using genetic rescue. Um, Joy Lanzendorfer, a very frequent Alta Journal contributor, wrote an article for Alta Online called Better Diversity Through Cloning, and it details the work being done by San Diego's Frozen Zoo, the world's largest biobank for living cell creatures. So what we're going to do today is talk about the science, the ethics, the possibilities surrounding Ben's work as one of the scientists pioneering the process. Ben is a lead scientist for conservation cloning projects at Frozen Zoo's partner Revive and Restore. And we're going to talk to him about their work, about that. A little bit more about Ben before we begin, because his bio is fascinating, and I encourage you to dig a little deeper online if you want. But briefly, Ben collaboratively pioneers new tools for genetic rescue and de-extinction. As a lead scientist, he heads Revive and Restore's de-extinction efforts and is the lead coordinator for conservation cloning projects. In 2022, he expanded his role to include program manager for the newly launched Biotechnology for Bird Conservation program. Ben's primary passion is the restoration of the extinct passenger pigeon, the goal of Revive and Restore's flagship project, the Great Passenger Pigeon Comeback. So, and there's much more to the work Ben does, but do you not have 20 million more questions just about that? We will try and get to some of those. Before we do, though, let me tell you about Alta. Alta Journal is an award-winning quarterly magazine and website focused to California and the West. This topic is California-y because the Frozen Zoo Bank is in San Diego, California. But Ben, you are not in California today. I'm going to ask you where you are in just a second. Alta, we produce free events like this that take you deeper into the articles from our magazine, our website that's updated daily. We also host the free California Book Club, which takes place monthly. We've got Danzy Senna coming up, Tobias Wolff coming up, really cool authors participating in Litquake events. So if you're interested in what we do, if you want to support really independent, thoughtful journalism, I really hope you will consider visiting altaonline.com, checking out the work we do, and supporting us with a membership, buying a tote bag, whatever you want. To join us as a member, it's $50 a year. You get 4 gorgeous issues of Alta, a little present in the mail. Currently, it's a book of the first 5 years of all of the cartoons that have appeared in our magazine. So think of us when you're shopping for magazines. There is a Q&A button at the bottom of the chat. If you've got questions for Ben, and I am sure you will, please use that to ask your questions there. There's also a chat, and we love to see where people are zooming in from. I am in Novato, California, just north of Restore and Revive's location in Sausalito. But Ben, you are not there. Where are you?
I live in a little town, Brevard, North Carolina. So that's where I'm raising my family.
Sounds fantastic. So I mean, the first things first, really. I realize that like cloning endangered animals is a really big topic, and we will dive into it. Before we do, what led you to doing this? What can you very briefly kind of take us on your journey from growing up to having a job that didn't exist, presumably, when you were growing up?
Yeah, I get asked this all the time, especially by students. I've spoken to students from kindergarten to, well, I guess everyone can consider themselves a student, but I've spoken to elderly communities everywhere, and everyone always wants to know how I got this and how I got interested. So really fast, like many kids, when I was a child, I wanted to be a paleontologist because I was obsessed with dinosaurs. I grew up in Western North Dakota. I didn't live somewhere to find dinosaur fossils, but I was finding Eocene age fossils where I was, well, Paleocene. And I was fascinated by that. And so that really got me gripped on extinct species. But I also grew up not very far from Theodore Roosevelt National Park, which I think for anyone involved in conservation, that really is kind of the mecca because Theodore Roosevelt is really considered what the kind of first president to really care about nature and the environment, and it was a huge turning point for our nation in terms of conservation history. And it was at Theodore Roosevelt National Park where his mindset about nature and preserving it for legacy in the future really took shape. And his journal articles there, entries there, really showcase that. And when I would visit the park, we would see bison and we would see bighorn sheep and we would see elk. And as I grew up, I learned that all three of those species had actually gone extinct in our area in the late 1800s and early 1900s. And from the 1960s to the 1990s was when people started reintroducing those herds of animals. And so I was exposed to the concept of restoration really early. And I was really fascinated by that. The wolf reintroductions of Yellowstone at the time in the late 90s and early 2000s were very controversial. The human genome was being sequenced. There was a lot of stuff going on that just made me think there's a huge power in the future of genomics and biotech. And I learned about more recently extinct species. I got hooked with passenger pigeons. We've got their images behind me. And I just thought to myself at age 13, I was like, I bet we could try to recreate extinct species someday and not just reintroduce something like a wolf where we go grab one from Canada and bring it down to Yellowstone, but actually recreate something that's extinct everywhere in the world and restore habitats that way. And I set my entire education trajectory based on that. And as I learned more about biotechnology and as I got offered kind of the job to work for Revive and Restore, which was through passenger pigeon interests, it became clear that the tools we're building to try and recreate something extinct could certainly be used to help tons of living species. And there's a huge need for biotechnology and conservation. So I've spent the last 10 years now on the job, literally conceptualizing all of this and hopefully rolling out some really cool stuff.
Okay. I need to just get this question out of the way because as many of us who lived through the 90s, my first reaction to reading Joy's article was, this is Jurassic Park. You are in Jurassic Park, for those that have seen the movie, they take DNA from a dinosaur from the blood of a mosquito that was trapped in a fossil, and they use that to recreate dinosaurs. Now, I realize the difference is you just mentioned it's not trying to clone or genetically kind of figure out extinct animals, it's endangered animals. But otherwise, what's the difference?
Well, I'll say a couple things first. Is that one? Yeah, I've been asked about Jurassic Park so many times over
I
the years
know, I
that
know.
I started to kind of really get frustrated with the concept of Jurassic Park. But I will say I've fallen in love with Jurassic Park again recently because I'm a well, I guess I'm not a new dad. My children turned 6 yesterday, and maybe it was too young, but I let them watch Jurassic Park, and they were just so awed by the concept of Jurassic Park as a movie, and they loved the movie, and it is such a great movie. It's a fantastic film. So I'm a big fan, but it had nothing to do with my interests in what we're doing, and the science is quite off, and the plot is quite off. So for anyone that really wants to delve into this, first thing is that Jurassic Park is actually a book about chaos. The concept of a dinosaur park was just contrived just to get across the idea that we can't control things and chaos theory exists. Um, that said, everything's contrived, right? They've got one computer guy who they don't pay enough, who then sabotages the park. They have no rifles at the park. They just have shotguns. I mean, like, they don't have ditches around their animals. Go to a zoo. In every predator exhibit is a giant ditch. That animal cannot escape its pen. Like, it's just everything about it ignores everything that people have been doing for many, many years. And it's to create that thrilling story of, oh my God, we're being chased
Yeah.
by dinosaurs. The science itself is kind of goofy. So they're talking about sequencing DNA from dinosaurs and inserting it into like frog DNA and things like
Yeah.
that. And that's not really how we would be going about doing this stuff. So first off, there is no dinosaur DNA. There have been claims about soft tissues in dinosaur fossils, and I want to tell everybody that's listening, I've worked with that personally, and it doesn't exist. So anyone that can go out and find a chunk of dinosaur bone or fossil, and this can be a chunk of fossil that is literally on the ground surface being rained on. It can have plants growing through it. You can crack that open and you will find these so-called soft tissues that look like blood vessels and osteocytes. And what those actually are bacterial endocasts. So bacteria has invaded the fossil and started growing on the insides of where blood vessels and cells were in the bone. And basically as they secrete minerals and other waste products, they create what looks like a little soft tissue, and it's very convincing. When you look at it, you go, "Oh my gosh, there's dinosaur blood vessels. We're going to get DNA," but there's nothing there. The proteins that have supposedly been claimed from dinosaur fossils all match proteins from living species, so they're most likely contaminants. It's not. We don't get anything from dinosaurs, and so DNA we can get maybe from something 1 million to 2 million years old if it's from the permafrost of Siberia. Most places in the world, 10,000 years is a pretty far stretch for being able to get DNA from something. So we can't get a genetic blueprint for a dinosaur, but we can for something like a passenger pigeon or a thylacine, um, anything sitting around in a museum drawer, we can get genomes from. They're highly fragmented, they're difficult to work with. And when we sequence that DNA, we're not getting an actual passenger pigeon piece of DNA that we then have the code for later. That molecule is practically destroyed when we do the sequencing. We get the digital code after everything's done. So it's just digital code. To recreate it, it has to be resynthesized in the lab and then inserted into the genome of a living animal. So that frog part is kind of true. You could synthesize a piece of DNA, put it into frog DNA. That's kind of ridiculous because even in 1993, people knew that dinosaurs were closely related to birds and crocodiles. Using frogs was nonsense. Um, but yeah, so it is kind of true in the fact that if you wanted to recreate what we're trying to do, like with passenger pigeons, we
Yeah.
would synthesize pieces of their DNA, and then rather than just insert them into a living pigeon's genome, we would rewrite that genome. So we would actually kind of cut out the pieces of DNA in the living pigeon and overwrite them with the passenger pigeon sequence. And that's pretty limited right now. We're working currently on seeing whether or not we can increase the order of magnitude in which we can do that. And I believe we're going to have really good luck with that.
All right, so let's dive into the species that it's possible to do because it's in reading Joy's article, I kind of like to do Alta Live, assuming that half of our audience has read the article and half hasn't. So it seems to me that there are only select animals with the frozen zoo, I guess, and your team is working with them, is trying to get as much DNA from extinct or I'm sorry, not extinct, but endangered animals as possible and banking it. So first question is, how do you decide which animals, in addition to carrier pigeons, the black-footed ferret, etc., how do you decide which animals to zone in on? How do you get that DNA and what happens to the DNA?
Yeah, so there's several layers to unpack here. First off, is that, as I said earlier, biotechnology holds a lot of promise for conservation. But it's still an emerging field, right? So we can't necessarily apply anything that works in a lab mouse to a wild species. There's a lot of research that has to happen. Um, and so for certain aspects, we look at this current state of the science. We work with black-footed ferrets because they have a very closely related domestic species, the domestic ferret, that is a biomedical model in research in which there's a lot of known knowledge. So we can use some of that knowledge to help figure out the biotech steps to use something like cloning or gene editing to help genetic diversity and facilitate adaptation in the endangered black-footed ferret. This is also a species that has been saved from the brink of extinction. And people have been breeding in zoos and other facilities very successfully for nearly 40 years. They've gained a lot of knowledge about the species ecology, its reproductive biology, its habits. So once again, we have a huge knowledge base and an infrastructure to work on that was built by hundreds of people over decades, who are really the real heroes that allow us to even begin to contemplate bringing in biotechnology. And that includes the conservationists at San Diego Frozen Zoo, who have been saving cell lines from these species for now since 1975. And there's a common thing we talk about DNA a lot, but it's really important to think of what's in the frozen zoo as cells. They're living cells. They have whole genomes inside of them. When you take them out of the freezer, you can grow them in a flask and they divide. They are a living entity. So the DNA is intact, it's alive, and that is the best thing to start with. If you want to reproduce an animal, you need to start with a living cell. Once something is extinct, like a passenger pigeon, if we don't have any living cells, then the work is tons harder to think of how you resynthesize a creature's DNA versus actually having it. So we have been, working with Fish and Wildlife Service and San Diego Zoo to really advocate the need to biobank those cells because we know there might be species that continue to go extinct before we can save their habitats, before we can convince nations to put more money into conservation efforts. We are fighting an uphill battle as a community because we live in a global environment that everything we do every day only exists at the exploitation of natural resources. Until we reach points in which food production, energy production, everything we do is sustainable and renewable and is cohesive with natural environments, we will continue to lose habitats and lose populations. But conservation has proven that if you put enough work into it, you can restore those things. The best thing to restore from is the living tissue itself. So we need to go out and get skin biopsies, feathers, things like that from endangered species, get them into the biobanks and cryopreserve them, because then in the future we can unlock biotechnology to help do that restoration, which is what we've been doing with black-footed ferrets. So we, with San Diego Zoo, Fish and Wildlife Service, and the amazing cloning scientists at Viagen Pets and Equine, took cells that were preserved in 1988 when I was 1 year old. Now everyone knows how old I am, or how young, depending on your perspective. And in 2020, took them out of the freezer, sent them to Viagen from San Diego, and used those to clone the first clone of an endangered species in the US, the black-footed ferret Elizabeth Ann. We've produced 2 more clones since then. We're continuing with the program. And what was so unique, and this is what kind of guides us in picking which species we kind of work with in the future, is that there was a clear need for what we're doing here to justify all the research and effort to start bringing in biotechnology. And that was the animals we've cloned are what we call new founders to the population. So really fast for anybody who didn't read the article and doesn't know, black-footed ferrets went extinct in the wild in 1987, the year I was born. And they, conservationists in Wyoming, brought in the last 24 animals that existed in the world into captivity to breed. Of those, a total of 18 survived. 14 of those successfully bred. And of those 14, 7 of them were actually the founders. So seven of them were the offspring of 7 others already in the wild. So every black-footed ferret alive today, there's about 600, 300 some in the wild, about 250 in zoos, are descended from just 7 animals. It's a tiny gene pool. Every black-footed ferret alive today is basically somewhere between a half sibling and a first cousin.
To every other black-footed ferret.
Yeah, yeah.
And this is what, when you mentioned genetic diversity earlier, this
Yeah.
is kind of what you were talking about. You're only dealing with these original 7 ferrets and all of
Yeah,
their grandkids.
exactly. And the cells at San Diego Zoo are from a female that was not one of those original 7. So cloning it and bringing it back to life today adds an eighth animal, an unrelated animal to the population. We sequenced her genome and analyzed it, and she has far more genetic diversity just in her own genome than any other individual living ferret has today. And so she's going to bring back genetic diversity. And diversity is basically the bread and butter of conservation and adaptability for these species. So we're really excited about that aspect in the fact that reaching back into the past like that with biobanking and a technology like cloning, that's the only way possible to bring back genetic diversity from the past that has been lost. If you're if you're not using biotechnology, and a great example of this that most people are aware of is the Florida panthers. Back in the 90s, Florida panthers, there were less than 30 of them. They were inbred so much that they were starting to see deformities, they were seeing sterility issues. And so the decision was made to bring in mountain lions from eastern Texas to boost the genetic diversity, which worked. They brought in 8 females in 1995, and the population of Florida panthers has actually been growing ever since. Their biggest challenge today is actually having enough habitat. It is no longer a genetic diversity issue. But
Are they
they
still
had,
Florida Panthers?
yes, yes. So there's about nearly 200 of those individuals living in Florida now. So it's from 30 to up to 200 in the past 30 years.
Even though
Uh,
they were bred with, oh, I'm sorry.
Yeah, so their genetics are mixed now.
Okay.
And so yeah, there are a couple subspecies now mixed together. The Florida panther genetics, it has been shown, have persisted. All of the living individuals today are still descended from Florida panthers. They just all carry some Eastern Texas genetics that helped infuse that population. That was only possible because there were other subspecies and populations of mountain lions around to get individuals and bring in and increase the genetic diversity. For a species like the black-footed ferret, where all living animals are descended from the same few animals, there's no other source to
Right.
bring in new diversity. And that's where biotechnology is essential. And we can build upon that to not only reach back and clone an animal that's new to the population from the past from a biobank, but now, if you think about the same technology we want to use to do something like recreate an extinct passenger pigeon, we can get into a museum drawer at the Smithsonian or the Museum of Southwestern Museum of Biology and open up a drawer and see a stuffed black-footed ferret from 1895 or from 1915 that represents a population that was completely extirpated, and we can sequence its DNA and figure out what regions of the genome have diversity that are completely lost today and gene edit those back into the population. So, there are more ways to explore how to get some of that diversity back. But as I said, if you don't have the living cells, it starts to become much more complicated. So those living cells biobanked are key, and expanding those biobanks are a huge mission for us to build upon what San Diego Zoo has been doing for 50 years.
Are there other biobanks other than the San Diego?
Yes, there's biobanks all over the world. There's actually a website you can get on, the Global Genome Biodiversity Network, GGBN, of 100 museums and zoos that are kind of a coalition that are working on expanding their biobanks. A lot of these institutions, they used to just preserve tissues in ethanol specimens or just stuffed, taxidermy, but they started to realize over the years that cryopreserving tissues in a particular way actually preserve them in a living state, and that would be more valuable. So there's a lot of institutions, the Smithsonian, the Burke Museum, the London Zoo now it has a stem cell lab. So, or is it, sorry, the London Natural History Museum. A number of institutions around the world are expanding into this space, but they all look to San Diego Zoo as kind of the gold standard of what to achieve.
Interesting. I want to talk about cloning and the possibility of being able to clone and reading Joy's article. I mean, I really encourage, if you haven't read it, read it. But I had so many questions about, wait, how do you do this? If you are able to, let's take the carrier pigeon, for example, as there's enthusiasm in the chat for
Endangered
brink.
pigeon.
I'm sorry.
Passenger pigeon.
Oh, I'm sorry, passenger pigeon. Okay, that's
Sorry, even ornithologists mix them up.
okay. Well, great, because I'm just a lady who sometimes looks at birds. But there is enthusiasm in the chat for bringing that back. So, okay, you've got this passenger pigeon DNA frozen now, alive, frozen. If you are able to clone pigeons, and reading Joy's article, I learned, just to briefly summarize it, totally tell me if I'm full of it, Ben, but it takes several generations of cloned animals before you can put them in the wild and de um what is that? De-extinction? No, de
Extinct their alleles.
yes, yes.
Yeah, so cloning, well, first I'll just say one that not a lot of people are aware of the rich history of cloning there is. And the technology really hasn't changed much since, well, in a simple manner, it hasn't changed much since the first animal was cloned, and that was actually 1957. That was the year my father was born. A lot of people think it was 1996 when Dolly the sheep was cloned,
Yeah.
but actually it's 1957, and the first animal ever cloned was a frog. Um, and scientists both in, I believe, Korea and the United States achieved cloning the same year. And it's always been basically the same. You take the genome, the nucleus containing the genome of the animal you want to clone, out of one of its cells. You take the egg cell from a surrogate mother and you remove its DNA. You then fuse in the new DNA, and this triggers that egg cell to believe it has been fertilized. So when we think of fertilization and natural conception, you have the egg cell, you have the sperm, they come together, the sperm enters, and it creates a full complement of DNA. So you get half your DNA from your mom, half your DNA from your dad. And the egg cell is half, the sperm cell is half. And when that sperm cell comes in and the egg cell goes, oh, now I have two sets of DNA, a full genome, it triggers the embryo formation. So people started to think back in the 50s, as early as actually before the 50s, and the first real paper on this goes to 1952. They thought, well, what if you just fooled the egg by giving it a full complement of DNA? And it turned out you could. There's a lot of complicated steps to the embryo formation, but the embryo does all that work by itself. So you bring in the DNA and you stimulate, and you have to grow that embryo to be large enough to then implant into a surrogate mother. The reason this started with things like frogs and salamanders is they don't have surrogate moms. They just grow inside of a jelly-coated egg, so they could just do that and watch them grow in a dish. In 1981 was the year the first mammal was cloned, and that was a lab mouse. And what made the history different up to 1996 was Dolly was the first mammal cloned from a cell from an adult animal, meaning she was cloned from cells taken from a fully mature sheep. They took a little bit of sample from the udder of a sheep and they used those cells. Up until that point, the mice, the sheep, the goats, the cattle, everything that had been cloned from embryonic cells. They harvested embryos that were somewhat developed. You could probably consider them fetuses, actually. And they would take a few of the cells from that embryo and then clone it. The reason that worked is because embryonic and fetal cells are already what we call mostly pluripotent. They're able to become an embryo and grow into other tissues. The reason cloning an adult animal was so difficult is if you take a skin sample from a sheep, that cell has an epigenetic imprint to be skin. It wants to be skin developmentally. So when it goes into the egg cell, it has to be reprogrammed. A lot of stuff has to go. And actually, for years, people thought it was impossible to do. But Dolly the sheep changed that for everyone. And the reason that was so significant, the reason it leads into what we're doing with black-footed ferrets, is that this frozen zoo started saving cells from endangered species in 1975. And nobody breeding an endangered species, and this will come to back to your original question, when you're breeding an endangered species, you don't just harvest embryos for research. Every baby could be critical to growing that population. So you only take cells from mature individuals where you can actually take a biopsy without harming the animal, or you wait until they die and you take it right after death. So all the cells in the frozen zoo are from adult animals. And until Dolly the sheep was cloned, cloning existed, but it couldn't work with those cells. And when you're doing a breeding program like that, even without a clone, as I said, every individual is important and valuable. And you need enough animals from the zoo to basically need, it's kind of bad to call it surplus because it's not, but you need to produce enough offspring to not only maintain the animals you have in your backup population in the zoo, but then be able to reintroduce some of those into the wild without risking extinction, right? So
Okay.
it works to have large numbers of offspring to get out there. So when we think of the animals we've cloned, a lot of work and effort went into cloning these precious animals that now have the genetics from the 1980s. Um, and so we would never release one of those clones to the wild where it could get killed by disease or a drought or a bad rainstorm. Any number of events could just kill that animal without it breeding. So to ensure that we can actually get those valuable genetics into the wild populations, we want to be able to release entire cohorts of animals that carry those genetics so that if a few of them get picked off by a predator immediately or die, some of them will actually live long enough to breed and keep passing that on. So we need to breed those clones for several generations in captivity to build up enough descendants from the new clones to have enough animals to put out into nature. But that's exactly the same consideration already happening with everything that hasn't been cloned.
Okay. This is fascinating. I'm sure you all agree. All right. So I'm going to ask you one more question on my behalf, then I'm going to get to audience questions. Um, are there, and I realize that this technology is constantly evolving by the day, certainly by the month. Are there ethical rules, laws around genetic rescue, around cloning that either exist in the kind of international community, or are there unspoken laws around it? What are the ethical ramifications of this?
Yeah, well, that by itself is a fascinating subject, partly because, if we think just of the Endangered Species Act as being a landmark bill passed by Congress that has shaped modern conservation, that was passed in the 1970s and 73. That was that was before most of this biotech stuff ever came on. None of the policies envisioned a future where we could clone something from the past. That bill was ratified, a couple years before the Frozen Zoo was even founded. So that stuff was emerging, but it was not on the minds of people at the forefront of conservation. Um, but as biotechnology for medicine and animal research and agriculture has advanced, a lot of laws and a lot of policies have been set in place that we know are at least good models from industry to begin starting to look at for how we would use for nature. Um, but it is unclear a lot of the time what we really should be doing from a policy level and a regulatory level. So we go back to our own ethics in the lab, which is no one can do research on an organism without institutional welfare approval. I've done research on animals in laboratories, and I can tell everyone the paperwork to get approved is lengthy. You have to have every contingency figured out on if an animal gets sick, at what point do you intervene? How do you intervene? What happens to an organism after the research is done? Are you rehoming? Are you sterilizing? Are you euthanizing? How are you euthanizing? How much space does the animal have in its habitat? Will the experiments cause discomfort or pain? How is this justified? Have you minimized the number of animals needed for this experiment? Um, can you replace them with in vitro studies? Like all this has to be covered at any research institution in order to proceed working with a living animal. And that living animal doesn't have to be a cute, cuddly ferret. That could be even just an insect. So you need those types of things in place. The the zoo community, all institutions have adopted these policy regulations, and those institutional regulations are actually mandated by federal law. So there is a nice background for going through the ethics. But when we think about, but that was all for laboratory animals, right? So when we're talking about releasing the descendants of clones into
Yeah.
nature, this is new terrain. And so I just want to put a shout out to Ronald Sandler and Samantha Wisely, who published a paper on the ethical considerations of cloning, and they use the black-footed ferret work that we do as kind of a case study. People can look that up because it really has to come down to similar questions of, is what we're doing, is the value of what we're doing, does it justify the effort, and does it justify the potential risk? Do we have risk mitigations for the individual animals? Do we have risk mitigations in place for the environment? But it's always an iterative process. No one's just going to create a genetically engineered, edited, or cloned or other biotech made animal or plant and just put it out into nature. Partly because that would just be silly. It costs money and resources to create these organisms. It would just be silly to throw it out in nature where things are wild. So it's an iterative process of, we examine these things in a laboratory setting, then a simulated wild setting. Then we go through things like soft release and controlled releases, limited releases to an experimental site. That's all in the future. So there's a lot to cover. That could be an entire
Right.
workshop.
No, this whole, topic is gigantic. So let's
Oh,
get
it's
to
huge. Yeah.
as many audience questions as we can. Danielle asks, what is the realistic timeline for achieving de-extinction of the passenger pigeon? How close are we?
All right, how close are we? So I'll be completely transparent with everyone. When we started this project in 2012 and announced it publicly in spring 2013, on a day I was having a bad hair day. So it haunts me to this day.
We've all been
Um,
talking about it.
I know. Um, we said we were going to accomplish it in 10 years. That was incredibly lofty because in 2012, basically none of the technology we needed to recreate a passenger pigeon existed. And today I'm happy to tell you that none of it exists still. but but that's a bit of a lie. So even in 2012, we had the building blocks of what could do this project. So people had created genetically engineered chickens using a certain technology of germ cell culture and germline transfer, because you can't clone birds. And if you want to know why you can't clone birds, go to Revive and Restore's website, reviverestore.org. Go under our programs, find biotech for birds. I made a 6-minute video, animations. It explains everything about the bird biotech. And because that's an issue for conservation with all birds in biotech, not just recreating a passenger pigeon. We know there's a blueprint for how to do it from chickens. CRISPR-Cas9 came on the scene in 2012, so we knew there was a path for gene editing. It just needs to scale up to the level we need. But as we've gone through, we also need stem cell technologies as well. So there's a lot of stuff we're working on, and I'm happy to say that we are working on all of those today. From 2012 to 2022, it was a nightmare trying to raise money for passenger pigeon de-extinction. We expanded greatly as an organization through our work with black-footed ferrets, through our work with the San Diego Zoo at Przewalski's horses. And today we fund and help facilitate nearly 80 projects around the world in about 20 nations, with universities and a lot of researchers. And that expansion happened with living species. It did not happen because our funders and donors were really interested in passenger pigeons. But in 2022, an anonymous donor has dedicated themselves to passenger pigeon de-extinction and bird biotechnology. And so after the first 10 years of just struggling to get the science up there and really just laying the groundwork of communications, I can finally say that I believe we are now on a 10-year track, because we have multiple projects up and running in parallel. And I believe we can start gene editing the cells of living pigeons to be more like bandtail pigeons, possibly in the year 2029. So that 2032 might be a year we actually have something living and breathing that more or less looks and behaves like a passenger pigeon. It's hard to say because I think the ground foundational technology will be able to do it by 2029. But then we actually start hatching birds. We have to see how well and how much farther it is to get those birds to be the right birds we need them to be. But it will happen in our lifetime. We will not be having huge flocks of birds in the wild in our lifetime. That will be a century-long conservation effort that goes to, has a legacy beyond. And that's not abnormal. I mean, we are still working on conserving bison today 120 years after that conservation effort started. Conservation takes time because humans have degraded habitats so much. We continue to degrade habitats and annihilate populations. This is the reason a good conservation effort to save a species like the California condor or loggerhead shrikes or American alligators, which are now successfully delisted, bald eagle is delisted, can take decades and decades.
Yeah.
But yeah, I think we're on track. So we have not updated our website yet. You can look at the passenger pigeon work on the website. We haven't updated it yet because we're going to update that more towards the end of this year. These new projects have just been launched. There's not a lot to show about them yet. And we're hoping, that we get a few nice things to show people within the next year.
Okay, Danielle, 10 years. There you go. To answer your question, get ready for that passenger pigeon to be sitting on your shoulder in 10 years. All right, we've got several questions about how long this living DNA that you're banking is going to last.
Theoretically, forever. So it's cryopreserved at -1, oh gosh, 196 degrees Celsius or something like that. I'd have to double check. Someone can probably Google me right now, but it's the temperature of liquid nitrogen. And basically at that temperature, the living functions inside the cell just completely stop. And so it's in a state of suspended animation, and that could last forever. No one really knows. So because these technologies, cryopreserving sperm or eggs or cells, only started in the 1930s to 1950s. The oldest biobanks in the world go back only to about the 40s or 50s. People don't typically reach back and use samples that old. Most things that have ever been cloned, or most things used for IVF, or most embryos transferred, have only been cryopreserved for a few months to a few years. I don't want to claim this absolutely, because I'm sure agriculture has beat this, but I think as far as the realm of conservation, the Przewalski's horse clone used cells that were 40 years old, that had been frozen for 40 years. When we cloned that with Viagen and San Diego Zoo, the black-footed ferret, the first time those cells were cloned, they were 32 years old. Those are both the oldest examples of cryopreserved cells for wildlife being used for cloning. So I can say that they'll absolutely last 40 years, but most likely much longer.
Yeah, that makes sense. We just don't know because we've only been trying for a certain amount of time. Got it.
You have to come back in 1,000 years and ask again.
Right. Yeah. And it's 1,000 years then. Okay, we're so over time, so I'm going to only ask two more, and I hope that the rest of you do research and check out Revive and Restore. I dropped the website in the chat. I also want to let everyone know we're going to send you links to all of this stuff, to Joy's article, to Revive and Restore's website, to anything else that comes up today. We're going to shoot you an email later this afternoon or this evening, so you can dig further on your own. Okay, Matthew asks, kind of specifically, is there any plan to bank all living species found in Florida Keys or other low-lying areas that are endangered because of climate change, or that could be endangered relatively soon because of climate change? Is that a factor in determining how you're going out and banking DNA in terms of prioritizing or triaging?
It is. I am happy to say that through our work in a pilot program at Fish and Wildlife Service and Viagen, Pets and Equine, and San Diego Zoo, we have preserved cells from two endangered Keys species, the Key largo cottonmouse and the Key largo woodrat. Currently, the biologists working in that area are trying to get tissues from the Keys marsh rabbit. There are a number of species that people are working with, beach mice from throughout that region, where we've gotten a few beach mice as well. One of the issues with any species for biobanking is we know we can biobank tissues with a certain method, but beyond mammals, we really don't know whether or not the methods we're using are preserving tissues and cells adequately. And beyond mammals, we really can't culture cells effectively for a lot of species. So like amphibians, we can't really culture their cells that great. We can't freeze their embryos. We have work ongoing for coral reefs because the keys are disappearing from sea level rise, but their reefs are also in jeopardy from climate change. So we funded work by Mary Hagedorn at Smithsonian that working with a company called Bioporic that have successfully frozen chunks of corals for the first time. And that's a technological breakthrough. You can see the publication online that makes it finally a possibility to get samples from the world's coral reefs and biobank them for the future. And we know that those restoration can work once the environmental problems are fixed. But there's more R&D to be done. So yes, we would absolutely love to continue working with our partners to prioritize species at climate risk, but a lot of these species need significant R&D, and that's where I have to make the plug that Revive and Restore is a nonprofit organization. We run on donations, and we are currently fundraising for such R&D, particularly in amphibians. We have a project slated with just an A-list team of partners at universities and companies to be able to try and create embryo cryopreservation and better cell culture and stem cell culture for amphibians. But we need donations to be able to make that happen. We are fundraising to be able to start biobanking corals and more species at scale. And if we can raise more money for biobanking those Florida Keys species and others, we actually would like to get cell lines and reference genomes made for every listed endangered species in the US. That's nearly 1,600 native species. We've gotten about 16 of those. So, we're 1% of the way there with our effort. San Diego Zoo has already gotten cells from a few dozen more of those in their tenured history. But there's a lot of species, plants and animals, where we need R&D, and we need the ability to go out and make that effort happen. The money isn't going to come from the government because, as we can see from the government, they've got a lot of their own other issues they're working on. So conservation right now really relies on individuals to contribute to nonprofits like Revive and Restore. So if you check out our website and you think these are cool missions, definitely help out, because not everything goes to a passenger pigeon project or a ferret. As I said, we have a lot in our wheelhouse.
I popped the donate link in the chat. All right.
Awesome.
And we'll include it in the email. Okay, last question. I've got to ask it. Janine also asked it, and it's would these methods theoretically serve to recreate one of the predecessors of Homo sapiens? Or how far away are we from doing, those of us on two legs? Could you do humans?
I will answer this academically, because I just want to preface this with, I do not support the recreation of extinct populations of humans for many, many reasons. One of those big reasons is just the fact that we live in a society today with wealth inequality, racial injustice, sexism, and gender injustice. I would like to see us figure out how to be decent human beings to all of ourselves, and also learn how to cohesively live with other species before we even contemplate the idea of bringing in a completely new, diverse group of human beings. I mean, how much of a travesty it would be to say bring back a Neanderthal into a world where we know there are people that will hate them simply just because they exist. When in truth, we are part Neanderthal, and it's likely that having Neanderthals and Homo habilis and some of those other species that have contributed to our genome back would probably help us as a society, give us new perspectives, etc. But as I said, we don't listen to the perspectives of everyone we already have. That said, human biotechnology is the farthest advanced biotechnology. So technically, recreating the genome of a Neanderthal would be probably the easiest thing to do, is in terms of de-extinction. Recreating one of those would not use cloning. It would most likely use something called stem cell gametogenesis that has not been done for people yet, but it's a fascinating technology that is being pioneered to try and help people with fertility issues, where you could take a piece of skin from a living woman or man, turn it into induced pluripotent stem cells, and then create sperm and eggs from that to create an embryo for a couple that can't conceive, for same-sex couples to be able to have their own biological children. So there's a lot of research going into that. It has been achieved in mice. San Diego Zoo is working on doing this for the northern white rhinoceros so they can recover that population from frozen cells. We would like to use this technology with all endangered species. We are working on stem cell projects in birds to be able to do this with birds. We would like to be able to do it with black-footed ferrets, and that's another reason to kind of get on board and donate to us, is that we would like to advance these stem cell technologies across wildlife so that there are even more options to do this technology better and really work the best we can with the welfare of the animals. Because for cloning, you have to get egg cells from a female, and for any woman out there that has ever gone through fertility treatment, we know that that is not a fun process. It's the same for wild animals. Getting those egg cells is not great. That's why most of the time people doing this research will actually wait until an animal dies and then just get the cells from them after that, because we're not causing the animal any pain that way, or discomfort. With stem cells, if we can just create egg cells, in a petri dish from a harmless biopsy, that completely changes the game. And so these are the types of things we really want to continue to be able to do.
It is always so much fun to interview scientists who are super passionate about what they do, because you're like a professor that's just got the whole class like wrapped. This is so interesting, Ben, and I'm so grateful that you've taken so much time to educate us and help us kind of wrap our heads around so many complicated kind of scientific questions that you are working on with such passion and enthusiasm. I'm really, really grateful to you for taking the time, and to our audience for the amazing questions and excitement around this. So thank you, everyone. Again, this will be recorded, posted online, send it to your friends. Check out Revive and Restore and the amazing work that they're doing. I do want to invite you all next week on September 3rd. We're going to be hosting Morbid Anatomy. It's a community, a website, a new book that's all about kind of embracing end of life and the inevitability of death. And we're going to talk to Joanna Eberstein about that. But in the meantime, a big thank you to Ben Novak. I'm so grateful that you took the time today. So thank you.
Yeah, thanks for the opportunity. This was great.
All right, take care, everyone. We'll see you next time. Bye.